Good afternoon. Today, I'm going to talk to you about my research investigating emerging and re-emerging viruses and how they intersect with climate change. And I guess one difference for me and a lot of the other people, I think a lot of the other stuff is focused on re-emerging pathogens. I'm going to talk a little bit about potential emerging pathogens. So 15 years ago, this was the list of known so this was the list of known filoviruses with all of, including Ebola and Marburg, deadly viruses, with all of them except for Reston virus being found in Africa. Reston virus, which is non-pathogenic, was found in Southeast Asia originally. This is the list of filoviruses today, so it's jumped from 6 to 16. And one big thing to know about this is that most of these viruses, all these viruses known in blue, have only been identified by sequencing. And so therefore, we know nothing about the potential pathogenicity of these viruses. Also, none of these, almost all of these viruses in blue have not been cultured, so it's difficult to study them. But some of the work I do is using the genetic sequences of these viruses, using a system called reverse genetics, to generate recombinant versions of these viruses. And this is done in the Biosafety Level 4 lab over at the Needle on the medical campus. And most of the work we do is studying 4 different families of viruses, the filoviruses like Marburg and Ebola, Henipaviruses like Nipavirus and Longivirus, arenaviruses like Lassa virus, and nairoviruses, namely Crimean Congo hemorrhagic fever virus. So in order to study the pathogenic potential of these viruses, we partner a lot with researchers at the Center for Regenerative Medicine, the CREM, also at the medical campus. And they specialize in taking human-induced pluripotent stem cells and turning them into specialized cells and organoids, cells from the lung, the brain, the liver, the intestines, et cetera. And so using these cells and organoids, we use a comparative virology approach and take our viruses that, for example, that are pathogenic to humans like Ebola and are non-pathogenic, for example, like Reston virus. And then we take viruses like Yavu virus, a new filovirus of unknown pathogenicity, and we compare the infection in these different cell types and organoids. And we use these to look and see whether these human cells can be infected, a major barrier to emergence of viruses and new species. We look at how these cells respond to infection to both identify the pathogenic potential of these viruses, as well as to identify pathways that are involved potentially in the pathogenicity, so pathogenic markers, and finally also pathways that may allow us to develop antiviral therapies. So lastly, now I want to bring this into climate change, like I mentioned at the beginning. So there's sort of 2 different ways that you can intersect climate change in infectious disease spread. So one is emerging viruses, so things like Yavu virus, which is this new filovirus found in Southern Europe, as well as Longivirus, which was recently the cause of agent a couple of years ago of 35 hospitalizations in China. These are new viruses that, because of climate change, their host species might be found in new places, and so might spread virus. And then, of course, which a lot of other people are going to talk about today, you have things where you're expanding re-emerging viruses. So Crimean Congo spreading from Southern Europe. Now these ticks have been found as far north as Sweden, and then Lassa virus, which is predicted to spread from Western Africa through Central and Eastern Africa.